So this provides a bit of an outline for where we're heading today. So we'll spend some time talking about proteins, carbohydrates, lipids, and then vitamins and minerals. As an overview here for some of these, for proteins, when we ingest these proteins will first be broken down into individual amino acids, and we'll see how that takes place in just a few minutes. And then once we get to the individual amino acid stage, then they can be absorbed. And then once absorbed, the amino acids can be taken up by the blood and distributed to the various cells of the body. And there they can be used, as you can imagine, for the production of new proteins. The amino acids might be used for energy production in some situations, as well as the amino acids can be used to produce some other nucleotides, some other molecules such as nucleotides. And when we talked about the synthesis of nucleotides, we saw, for instance, glutamate was involved, and there's some others as well. And then some amino acids, such as glutamate, again, can be used for communication. So, for instance, in the nervous system. So we have proteins, and again, the proteins will be broken down into individual amino acids. We also have carbohydrates, as we mentioned before. And for carbohydrates, typically we have a variety of carbohydrates that we ingest. So for instance, we can ingest plain old glucose. We can ingest fructose, right? The high fructose corn syrup that's in like all of our food. So we can ingest that. And those two are monosaccharides. We can ingest some disaccharides such as sucrose or lactose, which are disaccharides. And then we can have some fairly complex carbohydrates that we ingest, such as starch, as well as, plant matter has a lot of cellulose in it. And the cellulose, while we don't digest the cellulose, it does provide a lot of fiber for the digestive system. So in terms of digestion of the carbohydrates, we're going to be breaking these down, as we'll see in just a minute, into simple sugars. And then these simple sugars are what get absorbed into the small intestine and then transported to various parts of the body. And then once found in the cells of the body, there we go. They can be used, of course, to produce energy, right? So for instance, they can enter into glycolysis and be used for energy for the cell. They can also be used as building blocks in the synthesis of larger molecules. So for instance, we had talked about things such as proteoglycans and glycoproteins earlier this semester. And as well as they can be stored as glycogen. So for instance, if we don't need those glucose molecules right away, they can be stored again as glycogen. And then for the fats here, again, typically when we ingest fats, there's a variety of lipids that are present. Most of the lipids will need to be digested into smaller components prior to absorption. We'll see this in just a few minutes as well. And then once the lipids are absorbed by the small intestine, again, they can, in certain circumstances, be used for energy production. They can be used in the synthesis of larger molecules. Some of them can be used to produce things such as steroids and other communication molecules. And then, of course, the storage form of fats in our body is going to be in the form of the triacylglycerol. So that's an overview of what happens with the proteins, with the carbohydrates, and with the lipids. And so let's look at each of these in some more depth here, and we'll stick with the lipids now for this next section. So, as I just mentioned, the triacylglycerol is the storage form of fat. And if you think about this for a minute, if it's a storage form of fat in the body, as it turns out, it's also the storage form of fat in various other animals that we eat as well. So when we have a meal, the triacylglycerol is actually one of the major lipids that we acquire during ingestion. And so if you remember here, this is the structure of the triacylglycerol. So we have glycerol in the middle here, and then we have our 3 fatty acids that are attached to it, shown on the sides here. And so, again, we have a large amount of triacylglycerol that we ingest, but then there are some other lipids as well, and there can be a variety of other lipids. And a couple of these are cholesterol, as well as some phospholipids can be found as well, and then some other lipids also. But these are some of the major ones that are found. So we focus on the triacylglycerol for just a minute here. So when we see where this occurs, so let me step back a minute. So where does the digestion of triacylglycerol occur? Well, when we eat our, I don't know, cheese sandwich or something, or grilled cheese, right? A bit of the digestion will take place in the mouth and the stomach in terms of, where the lipids get digested. However, by and large, most of the digestion of the triacylglycerol is going to take place in the small intestine. Okay, so a little bit in the mouth, a little bit occurs in the stomach, but by and large, it's mostly in the small intestine where it takes place. You should definitely know that the enzyme that is involved in digestion of the triacylglycerol is this enzyme called lipase. And with lipase, we get a little bit of lipase produced in the mouth. So there's a lingual form. There's also a bit in the stomach. So there's a gastric form. But again, the major place where lipase is produced is going to be in the pancreas. And from the pancreas, does the food actually travel through the pancreas? No. No, right? But rather the pancreas is going to be secreting a number of digestive enzymes into the small intestine. So we have lipase, and what lipase does is it takes the triacylglycerol and it can remove up to 2 of the fatty acids from the glycerol. And what we end up with is 2 free fatty acids, one here and one here. And then we end up with also a monoacylglycerol, and more specifically, the fatty acid is attached to the 2 position of the glycerol. So it's the 2 monoacylglycerol that is left with here. So again, for the triacylglycerol digestion, it occurs mostly in the small intestine, a little bit in the mouth and the stomach, and it's carried out by the enzyme called lipase. So in terms of a couple of the other lipids I mentioned, so cholesterol and phospholipids, well, their digestion is going to occur in the small intestine. And here we have a couple of enzymes, a cholesterol esterase, as well as another enzyme called phospholipase A2. And as you can see here, the cholesterol esterase will cut off this portion of the cholesterol. And then over here, the phospholipase A2 will cut off a portion of the phospholipid. You don't need to memorize the specific structures here or what regions are removed. But you should know, nonetheless, that the cholesterol gets modified by this cholesterol esterase, and then the lipids, the phospholipids, get modified. They get cut by this phospholipase A2. Okay, so we have these enzymes that play a role in lipid digestion. And again, depending on the lipid that we're talking about, whether it's triacylglycerol or cholesterol or a phospholipid, there are some different enzymes that are involved. There is another aspect of lipid digestion that we need to think about. So when you take a bunch of lipids and put them in water, what happens to them? They congregate, exactly. So now, one of the issues that we have with lipid digestion, especially, after the stomach, so as things get mixed up in the stomach, what happens is the lipids tend to congregate, and we end up with these fairly larger fat globules that are produced in the stomach. Yeah. And so these larger fat globules need to, the issue with them is that these enzymes that I was just talking about really can't get at the fat molecules in order to digest them. And so what needs to happen here is the larger fat globules need to be broken apart before these enzymes can really get access to them and digest the fats. And so the solution to this is that we produce these things called bile salts. And bile salts, what they can do is they can interact with the large fat globules, and in doing so, they disperse the fats that are found there into much smaller fat droplets. And so now, by increasing this surface area that is available, this now allows the various enzymes that I just mentioned to get in there and to interact with the lipids and digest them. These bile salts that are produced, they are made in the liver. Okay, so important to note, the liver synthesizes bile salts, and then they then are transported into the gallbladder, where they get stored, and eventually they may be released. They will talk about how they are triggered to be released in just a minute, and they get released more specifically into the beginning of the small intestine. Yes, the ileum. So let's talk a bit more about bile salts here. So bile salts are actually derived from cholesterol. We won't go through the synthesis pathway here, but you should at least know that there's a link between bile salts and cholesterol. And notice here that they are amphipathic. What does that mean, by the way? Yeah, very good. So they're both polar and non-polar. So they have a hydrophobic region and a hydrophilic region. And that sort of makes sense here for what we know the bile salts are going to do, right? So on one hand, they need to interact with the lipids, but then on the other hand, they need to interact with, say, water molecules that are polar. So the bile salts are amphipathic, and here, in this example, this is cholate. There are several different bile salts. Again, you don't need to memorize the specific bile salt here, but I put this up as an example because you see both the hydrophobic region here and then the hydrophilic region over here. So we have the bile salts that get produced, and I mentioned to you that they get stored in the gallbladder. One question that relates to lipid digestion here, but also for carbohydrate and protein digestion as well. But one of the questions we should consider here is, well, we have these various things such as bile salts. We've mentioned the lipase that's produced by the pancreas. These are all being released into the small intestine. So the question is, are these always continuously released into the small intestine, or are they released just when the partially digested food from the stomach has entered in? Yeah, the latter here is the answer. So what happens now is, in order to conserve resources, our body secretes the bile salts as well as the pancreatic digestive enzymes only when the partially digested food is coming from the stomach into the small intestine. And so the question is, well, what exactly, how is this regulated, right? There must be some sort of communication that's going on between, say, the small intestine and the liver, the gallbladder, as well as the pancreas. And indeed, there is some communication that is taking place. More specifically, there is a hormone called cholecystokinin, which is abbreviated CCK. And now what happens is when that partially digested food enters into the small intestine, there are certain cells there that can sense this, and they release a hormone, this hormone, called the cholecystokinin. This hormone gets released into the blood, where it then travels to the pancreas as well as the liver, and once it's found there, there are certain cells in those organs that can bind to the hormone. And when this takes place, it triggers the gallbladder to release the bile salts. And it also triggers in the pancreas to release a variety of digestive enzymes, including the lipase. And technically, it's found as prolipase when it's released from the pancreas. The prolipase, by the way, we'll talk about this more a few slides from here, is an inactive form of the lipase that will get converted to the active form lipase in the small intestine. So now, once we have our lipids digested, right? So our triacylglycerol has been digested, the cholesterol has been digested, the phospholipids have been digested. Well, what happens now is they form these fairly small groups of lipids that are called micelles. And these micelles can interact with the cells that are lining the small intestine called enterocytes. And so this results in these cells taking up the lipids. The enterocytes actually will resynthesize the triacylglycerol. And then what happens is these cells take the various fats, including the triacylglycerol, the cholesterol, the phospholipids, and they package them along with a certain protein, apolipoprotein B48. And lo and behold, they end up forming these things called chylomicrons. And I think you probably at least heard of chylomicrons earlier this semester when you were talking about lipid transport. If you didn't, either way, they are still formed here. And from here, the chylomicrons get transported into the lymph, then into the blood, and then they can be transported to various parts of the body. For the bile salts, these don't get absorbed with the lipids per se, but what happens is later on in the small intestine, there's a mechanism for the bile salts to get reabsorbed. And so they typically get transported, over 90% of the bile salts get reabsorbed, and then they get transported back to the liver where they can then be reused. So that's what happens with the lipids. Are there any questions with lipids before we move on to proteins? Okay, so let's talk about what happens with proteins for a minute. And remember, our goal here is to take proteins and to break them down into individual amino acids. Protein digestion is going to occur in the stomach and then in the small intestine. So let's see what happens in the stomach here. And as we know, in the stomach, the pH is going to be quite low, right? So typically the pH will be pH 1 to pH 2. And the hydrochloric acid that is responsible for this low pH is produced by these cells called parietal cells. So the parietal cells produce hydrochloric acid, and this hydrochloric acid is helpful here because it helps to denature proteins. So in other words, it causes them to unfold. And then what happens here is there are also cells that line the stomach called chief cells. And chief cells produce an enzyme called pepsinogen. However, pepsinogen is the inactive form of this enzyme. When cells produce an inactive form of an enzyme that gets cleaved, that must be cleaved in order to become active, these are called zymogens. So, in the digestive system, there are a number of zymogens that are produced. why is this important? So for instance, the chief cells here produce pepsinogen rather than the active form pepsin. The pancreas produces prolipase rather than lipase. The pancreas will produce some other proteases, again, in their zymogen form. Why is that important? Well, right, so it's not going to be continuously active, and why is that important? What would happen to the cells that produce them if the enzyme was continuously active?
Get tired and they get broken down.
It would digest, right? So in a chief cell, are there proteins?
Yeah.
Probably, yeah. And so if we produce these enzymes that we're looking at here as the active form, well, they would just go ahead and digest the cell that just produced them. And that doesn't seem like a good system, does it? And so because of this, many of these digestive enzymes are going to be produced as zymogens. And so pepsinogen is one example here. And so pepsinogen gets produced by chief cells. It gets released into the stomach. And what happens now is the hydrochloric acid that is found here, the pepsinogen interacts with the hydrochloric acid, and it causes a change in conformation of the protein. So that way there, it can cut itself. In addition, when pepsin, the active form of the enzyme, is present, it can go back and also cut pepsinogen molecules that are found there as well. So the bottom line here is the pepsinogen in the stomach gets converted to pepsin, and then the pepsin itself cuts the proteins into smaller peptides. And there are certain amino acids near where the pepsin cuts preferentially. We're not going to go into that level of detail here, but we should know that the pepsin cuts proteins into smaller peptides. So, are we at the level of amino acids yet?
No.
No. So now what happens is, now this partially digested protein exits the stomach. Before we talk about what happens to the protein, the peptides, we need to talk about how that acidity from the stomach gets neutralized. Right? So we have our partially digested food coming from the stomach. The pH is quite low. How does the digestive system, how does the small intestine deal with that? Well, what happens here is the pancreas can release a large amount of bicarbonate ions. And the bicarbonate ions will neutralize that acidic pH. I could ask you a similar question that I asked earlier relating to the CCK, to the cholecystokinin. And the question here is, does the pancreas always secrete bicarbonate ions, or is it only when the partially digested food is coming from the stomach? Probably, again, the latter. And again, I could ask a similar question. Well, what type of communication is occurring between the small intestine and the pancreas? And as you can imagine, there's a hormone that is again involved here. And the hormone is called secretin. And so now when the partially digested food exits from the stomach into the small intestine, there are certain cells that sense this and release secretin. Secretin then travels in the blood to the pancreas, where it binds to its receptor and triggers the release of the bicarbonate ions. Okay, so now we see how things are neutralized here. In terms of protein digestion here, the CCK, the cholecystokinin, is produced sort of whenever food products are coming into the small intestine. So that will then secrete the release of some proteases from the pancreas as well. And more specifically, the proteases that are released in the pancreas are shown here. And again, they are produced in the pancreas in their zymogen form. So chymotrypsinogen, trypsinogen. Notice here, at least for some of these, the O-gen, pepsinogen, chymotrypsinogen, trypsinogen, this is the zymogen form of the enzyme. And when they get converted to their active form, we get rid of the O-gen here, so chymotrypsin, trypsin, etc. There are a few others as well. So the carboxypeptidase, elastase, these all have different specificities for different amino acids and different peptides. The bottom line is that by the time all of these enzymes are done cutting up the peptides, we end up with individual amino acids. One question could be, well, how do we convert the zymogen form of these enzymes in the small intestine to the active form? And what happens here is there is an enzyme that is produced in the small intestine called the enteropeptidase. And now, when our trypsinogen that is released from the pancreas enters into the small intestine, this enteropeptidase cuts the trypsinogen and converts it to its active form called trypsin. And now, once this trypsin has been formed, this trypsin is responsible for cutting the various other zymogens that are found here and activating them. Right? So now we end up with the chymotrypsin, we end up with elastase, we end up with the carboxypeptidase. And the trypsin also is involved in activating the lipase as well. Okay, and again, the net result here is the peptides get cut into individual amino acids. And from there are the various cells of the small intestine that have a variety of transporters in them that will then take up the amino acid and then transport it into the blood. We don't need to memorize the specifics on this particular slide here. Any questions? Are there any questions on what happens with proteins? All right, let's move on to carbohydrates then. And as you can imagine, the carbohydrates that we ingest involve all the way from complex carbohydrates to simple carbohydrates. But the goal of the digestion here is to produce the simple carbohydrates. So in other words, the monosaccharides. Okay, so we have our starches here just as examples. We have our disaccharides that we could ingest as well. And in terms of digestion, let's focus on some starch, what happens with the more complex sugars to start with. The digestion of the complex sugars is going to start in the mouth. It's going to take a break in the stomach, and then it will resume in the small intestine. The digestion of the complex sugars, such as starch, is going to involve an enzyme called alpha amylase. And alpha amylase gets produced in the mouth and released along with the saliva. And in addition, the alpha amylase also gets produced in the pancreas, where it can get released into the small intestine. Again, the enzyme CCK will be involved in helping to trigger the release of the alpha amylase from the pancreas. And what the alpha amylase does is it breaks 1,4-glycosidic bonds. And so it can go around this complex sugar and break these 1,4-glycosidic bonds. With that being said, once the sugar molecules get too small here, so namely, once we produce some disaccharides or even some trisaccharides, what happens is the amylase is not able to deal with sugars that are this small. And so because of this, we're going to rely on some other enzymes to get us down to the monosaccharides. Okay, so the alpha amylase breaks a variety of 1,4-glycosidic bonds. And then what we see is in the small intestine, there are also a variety of other, other enzymes that will digest these smaller carbohydrates. I don't want you to memorize this list here, but I do want you to know what happens with this one here called lactase. And in just a few minutes here, we will focus on lactase because this is going to relate to lactose intolerance. And so we'll spend a couple slides on that in just a second. But nonetheless, after these enzymes here have been able to get at the carbohydrates, we are now left primarily with a number of monosaccharides. So maybe some glucose, some fructose, some galactose. And it's these sugars, these monosaccharides, that can then get absorbed by the cells of the small intestine, by the enterocytes, where they can then be, again, absorbed into these cells and then eventually transported into the blood. Again, you don't need to memorize the details of this slide. So I mentioned lactase and lactose intolerance. And so what lactase does is it takes a disaccharide called lactose and it cuts this beta 1,4 bond here. And this then produces galactose and glucose. So that is normally what happens. However, with lactose intolerance, what happens is for either environmental or genetic reasons, some individuals do not produce lactase or they don't produce enough lactase. And so what happens now is obviously in the absence of this enzyme, they cannot digest the lactose. You wouldn't think that would necessarily be a big problem just based upon what I've said so far. But what happens now is instead of that lactose getting digested, instead it actually gets fermented by the gut bacteria. And so we can produce a large amount of lactic acid here. This results in an osmotic effect that results in a large amount of water then entering into the intestines. This then results in distension of the gut walls, which results in increased peristalsis. As things are moving faster through the intestines, this can result in poor absorption of what we've talked about, so fats and lipids and carbohydrates. And of course, the net result here is a very watery diarrhea. Okay, so this is what happens with lactose intolerance. What can be a cure for this? I don't know a cure, but what can help treat it? Lactate, which is, what? Sorry, what did you say? Diapers. Well, well, first of all, not drinking milk once one knows, or having dairy products, as well as, you know, there's some lactase that is available for ingestion. All right. Any questions on that? So in the last section here, I want to talk a little bit about vitamins and minerals, and this will not be super in-depth, but nonetheless, we should at least have a basic understanding of what's happening with some of these vitamins and minerals and why they are important. So let's start with vitamins. And by definition, vitamins are small organic molecules that are required for certain biochemical functions in the body. And as it turns out, they are often precursors to coenzymes. Also, by the strict definition, vitamins must be obtained from the diet. However, in reality, there are a few things that we call vitamins that are not technically vitamins because our body can produce them, at least in small amounts. But nonetheless, the strict definition of a vitamin is that they must be obtained from the diet. There are both fat-soluble and water-soluble vitamins, and you should have an idea, you should know which of these are fat-soluble and which of these are water-soluble. So for instance, vitamins A, D, E, and K are fat-soluble, and then the water-soluble vitamins are a variety of the vitamin Bs, so there are a number of vitamin Bs, as well as vitamin C. So let's look at several of these here. Vitamin A, so this is one of the fat-soluble vitamins. By the way, if we're dealing with a fat-soluble vitamin, where do you, for those that have a receptor in the cell, receptors in cells, where do you think it's going to be? Inside the cell or outside of the cell? So fat-soluble vitamin? Inside. Yeah, so just like we talked about for hormones, the receptors for the fat-soluble vitamins will be found inside of the cells. And there are three forms of vitamin A: retinol, retinal, as well as retinoic acid. And they can have a variety of important roles in the body. So for instance, vitamin A plays an important role in the eyes, right? So it's a cofactor for rhodopsin in rod cells. It can play a role in cellular differentiation and turnover. And as a matter of fact, some forms of retinoic acid, some modified forms of retinoic acid might be used to treat some cancer, can be used to treat some cancers. And then it can play a role in growth and remodeling of epithelial tissue. One thing that's not listed here is it also seems to have some antioxidant properties as well. Vitamin D here is not strictly a vitamin. So it's more like a steroid. So even though we call it vitamin D, our body is capable of producing some of it. So what happens here is we can actually, as I just mentioned, synthesize some vitamin D. And more specifically, the 7-dehydrocholesterol can, when it interacts with ultraviolet light, can be converted to this form here called cholecalciferol. So, you know how we talk about, needing some sunlight for calcium, to process calcium appropriately? Well, this is why. So again, the UV light hits the 7-dihydrocholesterol, converts it to this form here, the cholecalciferol. And then this cholecalciferol can get modified by the liver and kidneys to the active form of vitamin D, which is this 1,25-dihydroxycholecalciferol. You should probably know this name here for the active form of vitamin D. And when you look at the various vitamins that are available for vitamin D, you can see that they come in some different forms here. So the 1,25-dihydroxycholecalciferol, you can see cholecalciferol here being in a vitamin as well. And again, these can bind to their receptors, and what they do is they regulate calcium and phosphate metabolism to maintain proper bone metabolism. And again, there is some antioxidant properties here as well. Vitamin E, again, a very hydrophobic vitamin. It's a mixture of tocopherols. You don't need to memorize these structures here. Vitamin E serves as an antioxidant in membranes. And then moving right along here, there's vitamin K, which is also hydrophobic. And vitamin K serves as a cofactor for certain blood coagulation enzymes. Okay, so for instance, for someone on warfarin, what shouldn't they, well, they can, but what, this is a bit, you'll see this in the future, but what happens here is some of the leafy greens, they have a high level of vitamin K. And so now all of a sudden for an individual, say on warfarin, that's trying to monitor their blood clotting, what can happen now is if that individual normally eats, say, very low amount of greens and then gorges him or herself on those greens, that can actually impact the ability of warfarin to help provide anti-clotting activity. So vitamin K here does play a role in some of these blood coagulation enzymes. And then lastly here, what I'd like to do is at least get through the next couple of vitamins. Vitamin B. So there are a variety of vitamin Bs here. You should probably know that these ones are considered vitamin Bs, so riboflavin, thiamine, niacin, etc. And all that I'm going to ask you to know in terms of what they do is that they can serve as cofactors for various enzymes. And then lastly, we have vitamin C, ascorbic acid. We all know this as an antioxidant. You also talked about vitamin C when you talked about collagen synthesis, so it plays an important role there as well. And then lastly, there are the minerals. I'm not going to ask you anything on the minerals on the exam. But nonetheless, you should know that, we know things, right? So calcium's important. Sodium, at least some level of sodium and potassium, iron are all important as well. All right, I know the last few minutes were a bit quick, But if you do have any questions, feel free to come on up and ask. Otherwise, we are done for today. I hope you have a great rest of the day. Thank you.
That was very nice. Thank you.
You're welcome.